back for acquired immunity, right? So we're going to be talking about the B and T lymphocytes. We won't go into great detail on this, but we do need to talk about some of the key characteristics and how they are activated, and also how they, once activated, lead to the production of many copies, and ultimately fighting the infection or cancerous cells. And so let's start with the T cells, right? We've got two main populations of naive, right, unstimulated T cells. Largely, again, once they matured in the bone marrow and then processed at the thymus. Remember, one of the key things in the T cell maturation and B cell maturation is we randomly produce the receptors that will eventually bind the foreign antigen and lead to stimulation and activation. But one of the key elements is we have to remove B cells and T cells that have been generated with receptors that match self-antigens, right? So we have to undergo what's called self-tolerance. And that, for the T cells, happens at the thymus. The cells that are then released from the thymus are these naive CD8 and CD4 T cells. They will largely hang out in lymph nodes and wait to be activated. Again, we've removed the receptor types, the populations of cells with receptors that might match our self-antigens. Because again, that would be really bad, because then all of a sudden we get activation of our T cells in response to our own cells, and we're killing ourselves, right? So, if we're in a lymph node now, draw a lymph node here. Funky looking lymph node. Lymph nodes have sort of we'll have sort of our flow going out of the lymph node. Remember, the key thing with the lymphatic system is it picks up extra interstitial fluid and things flowing around in it, and carries it back to the bloodstream, right? But in the process, so this is a lymph vessel over here. There's another lymph vessel. This would be an inflow vessel here. This would be an inflow vessel here. So out here in the interstitium of the tissue, right? Especially in those tissues underlying potential sites of infection, like lining of the GI tract, lining of the respiratory tract, skin. We have a specialized antigen-presenting cell called a dendritic cell. So that guy's out here and looks like a starfish, kind of. Right? And those are the dendritic components. The dendrites are these sort of cellular projections of the membrane. And what it does is it really increases the surface area for making contact with these foreign antigens. Okay, so the dendritic cell comes across the foreign antigens, right? And what it's going to do is phagocytose them, right? I know it's kind of small, right? But it's going to phagocytose, right? It's an antigen-presenting cell. All antigen-presenting cells are phagocytic cells, right? Because what they do is they phagocytose the antigen, process it, digest it, and then present it on the surface using the MHC protein, right? And then, again, for antigen-presenting cells, that's an MHC2. So it's going to be internally processed, and then this red guy will be the MHC protein. It's got sort of a funky X shape to it that would have the antigen presented there at its surface, right, for the B and T lymphocytes. So this guy's actually going to migrate into the lymph vessels, back up into the lymph node, again, which is where we have the B and T cells. So this guy will present the antigen to the T cells here. And so which T cells are hanging out? Well, let's pick one. Let's do the CD4 T cell first. Right, the CD4 T cell, this guy will show up show the antigen. So this structure, I'm going to make it blue here. This structure is that highly variable region during production of the T cell populations, known as the T cell receptor. Right? So that's the guy that's specific for the antigen. But remember, the MHC has already grabbed it. Right? So we got to put our MHC over here. Again, here's our presentation. And then we've got to make our rest of our dendritic cell. Right? So there's our dendritic cell presenting the antigen, an MHC protein, to the T cell receptor. Once that happens, we get activation, and there are other components. There are checks and balances here. But that CD4 cell becomes active, and now what we're going to do is we're going to get, remember, it's called clonal selection. Some people call it clonal expansion, which is also true. But we select the one that's specific for, remember, there's a whole population of CD4 T cells, same for CD8 T cells. Only one with the receptor that'll match that particular antigen. So once there's a match, we're going to do clonal selection. We've selected the one that's specific, and we're going to clonally expand it, meaning we're going to make many, many, many copies. Right? So now we're going to end up with all of these CD8 cells with the matching receptor, right? 'Cause it's going to look like the original. And those guys are going to go off and do their job. All right. Now, one of their jobs is to co-stimulate the B cells. In other words, you need CD4, and remember, now they're called helper T cells, right? The CD4 subtype, once activated, produces helper T cells and memory helper T cells. The active helper T cells will go on and do a bunch of things. The big one being help activate the B cells so that the B cells can eventually divide and differentiate and produce antibodies. But they also go off into the periphery to release cytokines, chemical signals that activate the phagocytes and do all kinds of stuff. In other words, they're helping to sort of like make the infection happen, or make the immune response happen by activating portions of the innate system, right? Remember that interplay we've got between the two systems here, right? But again, maybe its bigger role, I don't know, that's probably exaggerating, but one of the key roles is to help activate the B cells, okay? Now, I can change this slightly, and we're going to make this a CD8 T cell, right? And we're going to get the same sort of pattern of, presentation by a dendritic cell, right? This matching with the receptor to the antigen, clonal selection of the one that matched, right? Division, differentiation, make many, many copies. But now we'd have our product here would be CD8 T cells. Well, CD8 T cells are what are called cytotoxic T cells once activated. And those guys go off and kill the foreign invader through a variety of mechanisms. Largely, again, by secreting things that poke holes in the perforin and granzyme to poke holes in the invading or infected cell and stimulate apoptosis. Right, so that's where there's these, in this scenario, these activated CD8 killer T cells will go off and fight the infection. Remember, in both scenarios, we produce memory cells, which don't do anything during this initial infection. What they do is basically go into storage, and 2 months, 2 years, 20 years later, those cells are, upon secondary infection, those cells will rapidly be activated. They actually generate a bigger response than the initial cellular response. There's a lot of things that happen that make it a better response to that secondary infection. And a lot of times you don't even notice the secondary infection because the memory cells take care of it before you end up with symptoms that you even notice. Okay, so we've got our CD4s and our CD8s. What about the B cells? Well, the B cells have receptors similar to the CD8 CD4 cells, right? So now we're going to call this a B cell, right? And it's got a receptor. But now in this scenario, it's still going to require antigen presentation. That is one of the keys of the B and T cells. They require antigen presentation. But in this scenario, the B cell also requires helper T cell input. So we've got to have a sort of an interaction here. Just sort of put some cellular proteins sort of interacting, and it's a very complex interaction. But again, this is a CD4 helper, right? T cell, and it's been activated already, right? It's been activated by exposure to the same antigen, or an antigen from the same, doesn't have to be the exact same antigen, but it has to be an antigen from that foreign invader. And they're going to agree that we're being infected, in which case this B cell is going to become activated, undergo clonal selection, and now what you've got would be called plasma cells. The daughter cells in this case are plasma cells. Well, the plasma cells, and they don't actually, I have to get rid of our receptor here, because they don't really serve the purpose of binding anything extracellularly. Right? Oh, and the other thing too about this, the naive B cell, is that this blue guy, yes, it's the B cell receptor, but it's actually an antibody. It's just sort of an unsecreted antibody. Okay, so the antibody acts as the receptor. And then what do we do once they become activated? Make lots of copies of that receptor, but now secreted, right? And so now we've got all this, right? So coming out of these plasma cells would be all of these copies of antibody, millions and millions of copies of antibody, which again are specific to that particular antigen, because remember, it has the same molecular structure as the B cell receptor. So the antibodies are secreted, and the antibodies do, and remember the sort of the ends of the Y here, the top of the Y, are the binding site for the antigen. And so that site binds to the antigen and can do one of three things. Well, it does, or more than one in some cases. It can opsonize the foreign antigen, right? Label bacteria or a virally infected cell as damaged and foreign, labeling it for the phagocytic cells to come in and take care of, or remember, or complement proteins to come punch holes in it. It can also neutralize. In other words, by binding foreign antigens, it limits their ability to do anything, to infect other cells or damage other cells, because it neutralizes them. And then one version of neutralization is specifically called agglutination, which basically means that it sort of like cross-links the foreign antigens, like binds up a whole bunch of, because it's got two binding sites, it can sort of link to two different microbes, in which case, and you keep like, extend it outward, basically what you're doing is linking all the microbes together using these antibody linkages. That's called agglutination, when it sort of like lumps them all together. Basically what it's doing is hold them down. Again, all the other chemical signals are being released. It says we have an infection, and here come the neutrophils, and here come the macrophages, right? And right, jump on the labeled opsonized microbes that are being held down, phagocytosis, apoptosis, kill the infection. Okay, I think that's pretty much all we need for B and T cell activation. One thing I do want to point out is that we talked about the MHC2 version here and presenting to B and T cells. When specifically the T cells move out to, remember the CD8 T cells moving out to, kill the foreign invader, they can also recognize cells that become cancerous. That's not a foreign invader, right? Well, any of our cells have that potential to start producing abnormal proteins. And you might say, well, how is it going to recognize that? Well, all cells produce what's called MH1 protein, the class 1 MHC protein, and they take samples of the peptides that they produce, and it show them on the surface of the cell. So in that way, a cancerous cell or a virally infected cell is taking internal samples all the time, even if it's healthy, before it's sick or infected, and showing those things on the surface. Well, if they're normal antigens, if they're self-antigens, then the immune system ignores them. If they are foreign antigens, either viral peptides or they are cancerous peptides, meaning we've made abnormal proteins that don't look like our proteins anymore, now they should match, right, our T cells that we're making in response to these, right? And so those T cells, specifically the CD8s, but again, the CD4s will release cytokines, so they recognize it too. But they will recognize the MH1 presented antigens of any cell in the body if those antigens produced become abnormal, right? So it gives us the ability to check the intracellular functioning and normal protein production of all cells all the time. But in addition, by using the dendritic cells, the antigen-presenting cells, we can actively digest them and present them to the B and T cells that are found in the lymph nodes. Right? So we've got a way to check all cells of the body, as well as respond to infectious agents that have been phagocytosed by our antigen-presenting cells.